operon system is of two types. One is called inducible operon, the other repressible operon. Inducible operon and repressible operon. In inducible operon, the presence of a chemical substance called inducer, brings about switching on of the operon. So here, in the presence of inducer, the operon is switched on. So protein synthesis is allowed to take place. Inducer will be almost always a substrate. You know that a substrate is a chemical which is supposed to be acted upon by the enzymes. So when the substrate is present, the enzymes are required. So in the presence of the substrate, the enzymes have to be synthesized. Therefore, the substrate functions as the inducer, allowing the synthesis of those enzymes. So this is called inducible operon. On the other hand, a repressible operon is one where a chemical substance called co-repressor brings about switching off of the operon. So this is for switching on, this is for switching off. This co-repressor will be almost always a product. A particular biochemical reaction takes place in the cell. A product is formed with the help of a group of enzymes. Once the product is formed, once the product is available to the cell, no more synthesis is required. Therefore, the system has to be switched off. So the very presence of the product switches off the system. Therefore, this is called repressible operon. For inducible operon, the example is lac operon. This is what Jacob and Monod showed in E. coli. And for repressible operon, there is a similar example from E. coli. It is called tryptophan, lac operon. Lactose is the inducer. Tryptophan, tryptophan amino acid is the co-repressor. So let us see how these mechanisms work. Now let us say this is the DNA molecule. In this DNA molecule, let us assume that in this region there are three structural genes. I have labeled them as S1, S2 and S3. So these are the structural genes. Lying adjacent to these structural genes is a portion of the DNA molecule which represents operator gene. So this is the operator gene. Lying adjacent to the operator gene is another portion of the DNA molecule which represents the promoter gene. So this is the promoter gene. For this promoter gene, operator gene and the structural genes, there is a regulator gene. So this is the regulator gene. So this entire system is called operon. The regulator gene by transcription synthesizes a mRNA molecule. This mRNA molecule by translation synthesizes a polypeptide and this polypeptide assumes the necessary structural modification to function function as a repressor protein. So this is the repressor protein. Attached to this promoter gene, you find molecules of RNA polymerase. These RNA polymerase enzyme molecules, if they are able to reach these structural genes, they can bring about transcription, translation, and result in the formation of the proteins. It is possible if this RNA polymerase is allowed to reach the structural genes. Suppose this repressor protein binds with the operator gene. RNA polymerase cannot reach the structural genes. It means the structural genes are switched off, so no transcription, no translation, no enzymes are formed. So if this repressor protein has to be prevented from becoming attached to the operator gene, then the repressor protein must be rendered inactive, and that is rendered inactive by the inducer molecule. So inducer will bind with the repressor protein. Repressor protein cannot therefore bind with the operator gene. Therefore, RNA polymerase is allowed. Hence, protein synthesis can take place. So let us say the repressor is inactive. Let us say the repressor is active now. Repressor is active. When it is active, this repressor reaches the operator gene. Binds with the operator gene. Therefore, the operator gene switches off the structural genes. Hence, there will be no transcription, no synthesis of mRNA, no translation. So there will be no synthesis of proteins and subsequently the enzymes. This is the situation when inducer is absent. For example, in lac operon system, the inducer is lactose, a disaccharide. When there is no lactose available to the bacteria, there is no question of breaking down of lactose. Therefore, the enzymes will not be produced. These enzymes will not be produced, so there is no lactose metabolism. On the other hand, if lactose is available to the bacteria, then the bacterial cell has to break down this lactose, and that breakdown requires 3 enzymes to be synthesized by 3 structural genes. So these enzymes are designated as Z, Y and A. Z, also called lac Z, lac Y and lac A. Lac Z is an enzyme called beta galactosidase. Lac Y is an enzyme called permease, and lac A is an enzyme called transacetylase. Galactosidase breaks down lactose. Permease increases the permeability of the cell membrane to lactose. Transacetylase transfers the acetyl group. So when all these 3 enzymes are available, the cell can break down, bacterial cell can break down lactose. So when lactose is available, what happens? The lactose molecule binds with the repressor. Repressor becomes inactive. So repressor cannot bind with the operator gene. If there is no repressor binding with the operator gene, RNA polymerase is allowed. So S1, S2, S3, the enzymes responsible, the genes responsible for the synthesis of these 3 enzymes are switched on. So those enzymes are synthesized and the cell can break down lactose. So this is when inducer is absent. Let us see what happens when inducer is present. This is the DNA. These are the structural genes. S1, S2, S3. This is the operator gene. This is the promoter gene. This is the regulator gene. Regulator gene synthesizes mRNA. This mRNA translates into a protein, and this protein becomes repressor. This protein becomes repressor. RNA polymerase, RNA polymerase. Now suppose lactose molecules are available. These are lactose molecules. So this is the situation when inducer is present. Inducer is present. When inducer is present, lactose molecule reaches the repressor attaches to the repressor at a suitable site, therefore repressor becomes inactive. Therefore, this repressor cannot become attached to the operator gene. Now it cannot become attached to the operator gene. Therefore, RNA polymerase molecules can freely reach the 3 structural genes. Therefore, these structural genes now enter into transcription, three different mRNA molecules are synthesized. These mRNA molecules undergo translation, three proteins are synthesized, and these proteins become the enzymes LAC-Z, LAC-Y and LAC-A. So all the 3 enzymes are available. All the 3 enzymes now start acting on lactose. So lactose metabolism begins, and the bacterial cell is using lactose. Suppose all the lactose molecules, including the one binding with the repressor protein, suppose all the lactose molecules are broken down by these enzymes, metabolized by these enzymes. No lactose here, no binding with the repressor, so repressor becomes active. Repressor binds with the operator gene. Operator gene now does not allow RNA polymerase, therefore the genes are switched off. No transcription, no mRNA, no translation, no enzymes. So this is how the lac operon system functions in the case of prokaryotes like bacteria.